In this video, we'll look at plant nutrition. Plants make themselves up from the nutrients that they obtain from their environment. And they make themselves up by taking these nutrients and generating the structures of a plant. And the structures of a plant are by and large made up of macromolecules. You learned about macromolecules in Biology 211. You learned that carbohydrates, or polysaccharides, are made up of monosaccharides. And you learned that the formula for carbohydrates was CH2O. And so then you know, whether you thought about it or not, that sugars are made up of just three elements: carbon, hydrogen, and oxygen. You learned that proteins were made up of amino acids. And whether you thought about it or not, you realize that you've got your amino group, and you've got your carbon chain, and you've got your hydrogen and oxygen components right in the basic structure of the amino acid, any amino acid. And most of the R groups contain these elements as well, but there is an R group that requires sulfur. So nitrogen, carbon, hydrogen, oxygen, and sulfur are all elements included in amino acids and thus in proteins. Nucleic acids, you learned in Biology 211, are made of nucleotides, and nucleotides have a amino group, right, or the nitrogen base, sorry. They have a standard sugar, and they have the phosphate attachment. So N-CHOP, nitrogen, carbon, hydrogen, oxygen, and phosphorus are all of the elements used to make a nucleotide and therefore nucleic acids. Lipids, you learned, are really long chains of carbon saturated with hydrogen, or when they're not saturated, double bonded, and then whatever bonds are left for these individual carbons to make are bonds formed with hydrogen. I include this oxygen here because at the end of a fatty acid you have an oxygen atom. Now, because of this relationship, because most of an organism is macromolecule, and macromolecules are made up of just these 6 elements, and almost all of the macromolecules are made up of just these 4 elements, you could say that we are 98% N-CHOPs, 95% hunk. If we ask, why is hunk such a large fraction of our elemental ingredient set, we look to a periodic table that has not just a X and a Y axis, but a Z axis. And along that Z axis, the vertical axis of this graph represents the electronegativity of these elements. You'll see that hydrogen, carbon, nitrogen, and oxygen are elements that are very electronegative. And this high electronegativity leads to stable covalent bonds. It's true that fluorine is exceptionally electronegative, but fluorine will actually not share electrons in a covalent bond, but it'll rip off electrons and does not act as a suitable component in the construction of complicated polymers. What are the other elements that we find in an organism, or specifically in a plant? I've shown you several metal ions over the course of the term: magnesium in PFK, manganese in adenylate cyclase, zinc in carbonic anhydrase. Although I didn't show you it, cytochrome C oxidase has copper and iron. You know that hemoglobin has iron. So in each case, you'll find these metal ions in the active sites of enzymes and proteins. And they confer the advantage for these proteins of being able to make very complicated sets of ionic interactions. And so, in general, the metals are used in living organisms as catalysts that are functioning in the active sites of enzymes. N, Ch, A, P, S are all found right here in the periodic table. Turns out that plants also contain quite a bit of calcium and potassium and chloride. And these ions, because they're always found as ions in plants, are used for purposes that we've studied quite a bit. We studied them when we looked at stomatal opening and closure. We studied them when we looked at Pulvini cells. So these ions are used calcium as a signal molecule and potassium and chloride as osmoticum regulating ions. So if we look at all of the elements known to be needed for a plant, we find that there's N-CHOPs, which make up the macromolecules. There's these elements that are found as ions, which are mobile and used to regulate membrane potential. Well, so does protons. And more importantly, for these three, osmotic potential. Although remember, chloride's very important for maintaining membrane potential during stomatal closure. And then there's all of the metal ions, which are used primarily for catalysis within proteins. And these are all of the elements we know to be necessary. So many experiments have been done in which plants are grown not in the soil, but on media in which all of these components are taken away. And when you take any one of them away, the plant can no longer survive for more than a generation. Survives for less than a generation because the plant probably already possessed sufficient quantities of all of these elements.